West Nile virus. For a long time, we thought of it as a concern for places like North America or Southern Europe. But what happens when a pathogen you've studied becomes a new local threat? Well, today, we're diving into exactly that, the arrival and management of West Nile virus, right here in the Netherlands.
Just so you know, we've put this briefing together using a Gemini deep search, pulling in all the latest data and guidelines. And if you want to dig into the primary sources yourself, which I always encourage, you'll find a complete, detailed list in the linked Notebook LM document.
Okay, here's our game plan. We'll start with the new reality of West Nile virus here at home. Then we'll get into the nitty-gritty of the viral blueprint. After that, we'll cover the global consensus on care, the ongoing search for an antiviral, and finally, we'll wrap up with the key takeaways for your practice. So, first up, let's talk about the new local reality. The fact is, West Nile virus has arrived in the Netherlands, and it's here to stay. 2020, that year is a huge marker on the timeline for us. It was the first time we had confirmed local transmission of West Nile virus. This means it wasn't just an imported case. The virus is now actively circulating right here. And here's a really crucial detail. The strain we're seeing is Lineage 2. Now, this is significant because historically, this lineage was found in sub-Saharan Africa. Its arrival here is part of a much bigger story, its expansion across Europe, signaling a real shift in the virus's geographic footprint. Okay, so let's pop the hood and look at the viral blueprint. To really understand West Nile virus, you have to appreciate the elegance of the Flavivirus genome. The genomic strategy here is just a masterclass in efficiency. It's a single-stranded, positive-sense RNA genome, which means our cells can read it directly. And the entire genetic code is essentially one long sentence, a single open reading frame. The host cell's machinery translates this into one giant polyprotein. Then, both host and viral proteases, like the virus's own NS2B, NS3 protease, act like molecular scissors, chopping it up into 10 functional components. You get 3 structural proteins to build new virus particles and 7 non-structural proteins to run the replication show. It's this process, especially the viral protease and polymerase, that's a prime target for antiviral research. This brings us straight to the clinical heart of the matter, the global consensus on treatment. And spoiler alert, it all comes down to supportive care. So, this is the question we all face in the clinic. You have a patient with a confirmed West Nile virus infection. What's the recommended antiviral therapy? What do you prescribe? And the answer from every single major health organization is unambiguous and consistent. There is no specific antiviral therapy recommended for West Nile virus. Period. That is the bedrock of all clinical management today. And just look at the incredible alignment here. Our own RIVM, the WHO, the CDC over in the US, they all sing from the exact same hymn sheet. Management is exclusively supportive. Yes, drugs like interferon and ribavirin have been looked at in studies, but none have ever shown a conclusive real-world benefit. And this is so important to emphasize. This principle holds true for every single patient, regardless of age. The management strategy for children is identical to that for adults. There are no special pediatric antiviral therapies. The focus is 100% on supportive care. So, the fact that we don't have a specific treatment naturally leads us here, to the ongoing search for an effective antiviral and how we actually approach that in the lab. Now, for anyone involved in drug discovery, this is a key concept. How do we screen potential candidates? The gold standard metric is the Selectivity Index, or SI. It's a really elegant ratio. You simply divide the concentration that's toxic to host cells, the CC50, by the concentration that's effective against the virus, the EC50. A high SI is what we're all hunting for. It means the drug is a potent virus killer at doses that are perfectly safe for the patient. That's the therapeutic sweet spot. So what does this look like in practice? Well, you start with an assay, something like a plaque reduction neutralization test. This tells you the EC50, the concentration that inhibits 50% of the virus in cell culture. Then you run parallel tests on uninfected host cells to find the CC50, the concentration that's toxic to 50% of those cells. Finally, you just do the math. It gives you a clear quantitative number to decide if a compound is even worth pursuing. Of course, it's not just about the drugs. A massive area of research is looking at the host. Why do some people get terribly sick while others have no symptoms? It turns out our own genetics play a huge role. studies are homing in on genes like OAS1, which is a key player in the interferon pathway. It seems that certain polymorphisms or variations in this gene can make some individuals much more susceptible to severe neuroinvasive disease. So the story isn't just about the virus, it's about the host response. All right, let's pull all of this together and focus on the key takeaways for your day-to-day practice. So, what do you need to remember? First, West Nile virus, specifically Lineage 2, is established here. It's an endemic pathogen now. Second, the universal guideline is supportive care only. There are no recommended antivirals. Third, that rule applies to everyone, children and adults alike. Fourth, in the lab, the hunt for new drugs is all about finding compounds with a high selectivity index. And finally, remember that host genetics, things like the OAS1 gene, are a critical piece of the puzzle in determining who gets severe disease. And that leaves us with a big forward-looking question. We have to accept that West Nile virus is now a permanent part of the European virome. So the real challenge is this. How must our diagnostic and surveillance strategies, everything from our molecular detection in the lab to mosquito control in the community, how must they evolve to effectively manage this threat right here on a local level? Thanks so much for tuning in. If you found this briefing useful, please be sure to subscribe for more. We genuinely love to hear from you, so leave your thoughts, questions, and feedback in the comments below.